Hi, Bio20, 9.2, more detail of how the nephron functions to clean your blood and form urine. So it's a very interesting statistic that your entire volume of blood, which is 5 liters, is filtered by your kidney like 65 times a day. And that is a lot of cleaning. So that makes your blood always ridding those urea, uric acid, and ammonia, toxic substances, and keeping your blood fresh and clean. So continually, though, your cells are metabolizing food even when you're not eating because they're stored, and so there continually are wastes being formed, so your nephrons really never get a break. The thing about this is that when all that filtrate goes into the kidneys, we do not want to lose something very vital to our body, which is the water. And basically 99% of the water that gets squeezed into that nephron is going to be reabsorbed. So that's a big part of what we talk about, is the conservation of water in the filtration process. And so here you see that when you eliminate 1.5 liters of urine per day on average, you're going to have 180 liters that have gone into your kidneys in that 24-hour period, and 178.5 of that filtrate is reabsorbed as the kidneys are functioning. So that's a pretty amazing feat. So 4 stages are going to be talked about over and over. So we repeat the 4 major steps in formation of urine. And the first one, this is a very, very simplified photograph of your nephron in yellow here. But the first thing we have to do is create this filtration or filtrate by having blood go into very high pressure situation, which is very tiny little vessels, and that will force the filtrate into the Bowman's capsule. The next job that will be done is to keep the good stuff. And what I mean by that is we reabsorb anything we do not want to lose into our blood vessels. And so that would be actively transporting back all those food molecules that you just spent a lot of energy digesting in your digestive tract. So amino acids and glucose, for example, are going to be reabsorbed into the bloodstream. Next thing that will happen is any other bad guys, urea, uric acid, ammonia, or even some medications, penicillin, things like that. Those will be secreted into the nephron to get rid of. And we also have this very important job of reabsorbing water, which happens in several different locations in the nephron. So these are the 4 major events that happen. We'll outline where this all happens in the slides to come, but we need to filter the blood, reabsorb, keep the good stuff, get rid of the bad stuff. That's what secretion means. And keep our water. 99% of our water is going to be kept in the body. If you were not having this reabsorption happening, you would dehydrate very, very quickly, and that is a life-threatening situation. Okay, so filtration is interesting. That's like 20% of the blood components going into the Bowman's capsule. Remember, the blood is going in through the renal artery, and it is flowing through these very, very, very tiny capillaries. And when those capillaries turn so tiny, that causes a lot of pressure, and that pressure will push out 20% of the blood at all times. And what gets pushed out is called filtrate, and the filtrate does not have blood cells. Basically, looks like blood plasma, kind of clear, but it contains some good stuff and some bad stuff. And the pores there in the glomerulus, in those little blood vessels, are small enough that they do not let the blood cells go through. We don't want to lose blood and have blood in the urine. So that's one of those things that if there is blood in the urine, there is an inflammation or infection of the kidney, because normally the blood cells do not go through. There is high blood pressure, and but it's pressure that will just push the filtrate and its components in. If someone has high blood pressure as a medical condition, that could end up causing damage to those nephrons because it's already high pressure in there. So what happens is the filtrate collects in the Bowman's capsule, the very first part of our nephron. So those little blood vessels go in, and the Bowman's capsule is collecting that filtrate, and it's got some good stuff, glucose, some salts, amino acids, but also it's got some not so good stuff like urea, uric acid. Those bad guys are in there. So the components in there have to be dealt with. So the first thing we do with them is we do reabsorption. And it says that 99% of the filtrate actually does get reabsorbed while passing through the proximal, that very first tubule, and in the loop of Henle and in the distal tubule. 99% of our fluids, of our water in particular, get reabsorbed. And that's because we've got these microvilli that increase surface area, so reabsorption is very easy to do. And so if we look at the proximal tubule, these are highlighted in green in these little diagrams, and you can highlight them in your notes if you have printed those notes out. But basically, there's a lot of mitochondria at work here, and in the proximal tubule, that first section of the nephron is, well, I guess second section, the Bowman's capsule is where we collect the filtrate. So this second section, the proximal tubule, has a lot of mitochondria, and so ATP are used to keep the good guys. And keep the good stuff is how I like to just summarize that. We're going to keep things like essential ions, glucose, and amino acids that you have just digested. You want to keep those in your blood. So what's going to happen here is that all the good stuff is going to get, with active transport, put back into your blood vessel that's, I meant to be red on that one, blood vessel that's traveling around here. We're going to reabsorb all of those good things back into our blood. And then we move down to the loop of Henle, and you can see that it's the descending loop first. And here it's very permeable to water. Now I kind of need to look at the ascending loop information at the same time, because what's happening in the ascending loop is that reabsorption of sodium is happening. And so what that means is that sodium is going, salt is going, and then chloride follows, is going back into the bloodstream. Now you have to realize that blood is all around that loop of Henle. But if sodium is getting reabsorbed back into the bloodstream, that means that the blood essentially is very salty in that area. So it's very easy for water to be reabsorbed into the blood because the blood is salty, and water is going to go try and dilute that salt. So this is basically how reabsorption happens. Now, why is it more permeable to water on this side and more permeable to sodium on this side? I can't really answer that question, but this is how water reabsorption happens in the loop of Henle. It is the right side here, the ascending loop, that is bringing back sodium, and it is the left side here, the descending loop, that's bringing back water. You can read about the camel and how it has a very long, much longer loop to keep more water, as much water as possible. And we're going to go now to the distal tubule, the ones more distant. Remember, this was our Bowman's capsule. This was our proximal tubule. This was our loop of Henle. This is our distal, or more distant from the start. If we say this is the start of the nephron, this one is more distal. So this is the distal tubule of the nephron. And what happens here is that sodium is going to be reabsorbed again. Sodium goes back into your blood. chloride follows to make sodium chloride, and of course water goes in. As well, we have some secretion going on there, and secretion means that bad guys are going to be put into the distal tubule. So tubular secretion at the distal tubule means that any remaining bad guys, urea, uric acid, or ammonia, that are still in that blood that's floating around. Remember, we have a big capillary network here. Any bad guys that are floating around your nephron are going to be secreted. That means pumped into the distal tubule so they can be eliminated. Okay, that could be also, I mentioned this earlier, could also be any other toxins like medications, prescription medication bits can also be secreted at the same time. Okay, we are now left at collecting duct, our final step here. And collecting duct, you might have guessed it, what we want to do is concentrate the urine by reabsorbing a whole bunch of water. And so this spot though is under hormonal control. So you've got hormones that will say reabsorb lots of water, or hormones that say you've drank so much water today that you can release a lot of water. But we are going to see water reabsorption here in the distal tubule, in the collecting duct, and that is depending on your water consumption. It says here that when the filtrate comes out of this collecting duct, it is 4 times more concentrated than when it comes in. So it is definitely experiencing a lot of water that's being reabsorbed into the surrounding blood. All right, just a recap on this diagram. I just highlighted the yellow to show you where the nephron is, the red to highlight where the blood vessels are, and I'm just going to review that. I've put in maybe 3 or so diagrams into your notes to show you different pictures of the nephron, but they're all telling the same story. Filtration is happening here where the blood is going into the Bowman's capsule. The word efferent and afferent, A becomes before E, so the little tiny arteriole going into the glomerulus is called afferent. Glomerulus is where filtration happens. The one going out, the little arteriole, is called efferent. So if you see those words, that's what's happening there. Filtration is happening. We're pushing out 20% of the filtrate, of the components of the blood, forming filtrate in the Bowman's capsule. The proximal tubule, remember the first biggest thing to do is keep the good guys. And so we'll keep all the amino acids and the salts and glucose. And down in the next in the loop of Henle, we're keeping salt over on the right side, we're keeping water over on the left side, and that is moving into the bloodstream. At the distal tubule, we're going to secrete, get rid of any other bad guys in the blood, urea, uric acid, ammonia, and medications. And then over here in the collecting duct, of course, a whole bunch of more water reabsorption. So this is the same recap as what our notes were previously. These pictures show you the exact same sequence of events, as well as this review chart. And in terms of that urinalysis, which was, we did use simulated urine, but just for your entertainment, I will tell you that every time we did a urinalysis, I would always tell my class that it was kindly supplied by Mr. Preslav. And so then I'd have him walk in and say, did you find any diseases and disorders in my urinalysis? Anyway, sadly, we don't get to do that, but I did put 3 different semesters worth of urinalysis data on my slides, so you can check